Hi, good morning to all of you. A very happy Sunday. So today we are going to discuss another unit, that is molecular basis of inheritance. So there are three chapters in the second unit, genetics. One is what the experiment conducted by the Gregor Mendel and its details, and even post-Mendelian experiment also included in the chapter. So compared to this chapter, it is simple. And another one is evolution. And the most important chapter in this unit is what the molecular basis of inheritance only. So if time permits, we will go in detail about the experiment conducted by the Gregor Mendel. Also, we'll discuss some of the important points in the chapter evolution. So let us see the details of what the molecular basis of inheritance. Right. Come to the genetic materials. All of you know genetic material only responsible for the characters in an individual. So we are getting the genetic material from our parents. 50% genetic material from our male parent and 50% from the female parent. While discussing about the sexual reproduction in plants and animals, I told you very clearly that what you can say, the real process of fertilization. So during the fertilization, this genetic combination of these two parents only happening. And from that zygote only the entire individual is developed. We can find so many character in our bodies, like what our mother. We can find lot of character in our body, maybe inherited from our father. At the same time, we can find our own body characters also. So that is what the beauty of sexual reproduction. See the characteristic features of the sibling of the same parent, they are not alike. So these variations are very common only in sexual reproduction that ultimately lead to the evolution. That only nature required. That is why this nature is supporting sexual reproduction than what the asexual mode of reproduction. At the higher level of individuals, both plants, animals, and even these microbes also going for the sexual mode of reproduction, very primitive type in case of microbes. So let us see some of the what you can say facts mentioned here in the textbook related to what the genetic material, that is what the nucleic acids only. You know, nucleic acids. It is one of the important biomolecules only. There are two types of nucleic acid present in the living organism. One is DNA and another one is RNA. DNA is deoxyribonucleic acid and RNA is a simple ribonucleic acid only. See, there are two types of nucleic acid present in the living organism as a genetic material to express the character. And DNA, that is deoxynucleic acids, the most of the organisms are having the DNA as a genetic material, whereas RNA is also there, but only in retroviruses. Retroviruses like AIDS virus. It's having what this RNA as what the genetic material, it is what ribonucleic acid. But why most of the people, most of the individuals are preferring the DNA as a genetic material? Reason is very simple. DNA is more stable than RNA. Genetic material should be stable. It should not change its chemical nature according to the climate or according to what the physiological aspects of the individual or according to the age of the individual. It should be stable somewhat. So that is why this all individual except the retrovirus using the DNA as what the genetic material. But DNA can undergo very slow changes, that only making variation. We can find the difference in the body character, but it is very, very gradual and very slow. And it is because of what the changes happening in the genetic material DNA. And DNA can store information about the character of the individual. And this DNA is made up of two anti-parallel strands. Both are complementary. So that structure only we are going to discuss in this chapter in very detail. The structure of DNA and structure of RNA, then we will compare the differences, we will discuss, and we will find why this RNA is not selected by the nature as a genetic material. RNA only formed in the living world initially as a genetic material, but later, because of what its disadvantages, it is replaced by the DNA. Right. Then come to the DNA. It is a polymer of deoxyribonucleotides. Polymer, a long chain of deoxyribonucleotide. We will see the structure later. Deoxyribonucleotide is nothing but a complex molecule consists of what? Pentose sugar. That is deoxyribose sugar only in case of DNA. Then nitrogenous base and a phosphate molecule. This combine together to form the deoxyribonucleotide, and this nucleotide, deoxyribonucleotide, bind together to form a very long chain. Such a two chains are there in case of DNA, and these two chains are anti-parallel. One is running from 3' 5' direction and another from the 5' 3' direction only, and it is anti-parallel. So length of the nucleotide depend on the nitrogenous base pairs. Take the DNA, the length of the DNA depend on the number of nucleotide, and it depend on the number of nitrogenous base attached to it. The double stranded structure, which are anti-parallel, as I told you, it is running from 3' 5' direction, and another strand is in the opposite direction only, from 5' to 3' direction. And the haploid content of DNA in the human cell is 3.3 into 10 raised to 9 base pairs. Base pair means we can find later, we will see that guanine bind with the cytosine and adenine bind with the thymine. Such type of base pairs are there in the DNA in a haploid content. Normally our cell is diploid, so the total content of the DNA is double. 3.3 into 10 raised to 9 into 2. So it acts as a real genetic material for all the living organism except retrovirus, which is the genetic material of the retrovirus. It is not DNA, it is RNA only. It was evolved later after the RNA. Actually, RNA only produced in the living world initially as a genetic material to express a character, but it is later, what you can say, replaced by the DNA, because nature select the DNA because of its advantages. That is why most of the individual except the retrovirus are having DNA as a genetic material, and the RNA is unstable and it is unistranded. It's made with only a single strand and it is not very stable. So that is why these RNA viruses, keep on changing their characteristic feature within a short period of time. We used to tell it's what go for the mutation and changing the character, right? Because it is what the unistranded and it is what unstable compared to the DNA. Then this is what the structure of nucleic acids. Now we are discussing about both DNA and RNA. We will discuss both together. And while discussing, we will find out the differences in their structure as well as what you can say, arrangement of these molecules. So it is mainly made up of three components, both RNA and DNA. It's made with three components. One is nitrogenous base. Maybe heard about this nitrogenous base, nitrogen-rich base pairs only. And it includes what? Guanine, thymine, adenine, cytosine, and one more, uracil. So these are the 5 nitrogenous base present in the nucleic acid, both DNA and RNA. But in RNA, only 4 nitrogenous bases. That is adenine, then guanine, cytosine, and uracil. These are the 4 nitrogenous base in RNA, whereas in DNA, instead of uracil, thymine is there. And because of the thymine only, this what DNA show stability. And it consists of what? The thymine, then adenine, guanine, and cytosine. And addition to the nitrogenous base, pentose sugar is also there. Pentose sugar means what? The sugar molecule consists of only 5 carbon atoms. Pentose sugar. And there are two types of pentose sugar. In case of DNA, it is deoxyribose sugar, whereas in case of RNA, it is simple ribose sugar only. And we will see the details of all the things later. And the phosphate group, it is common in both DNA and RNA. Right. Then we will see one by one in detail. First, this nitrogenous bases. See, nitrogenous bases mainly categorized into two groups based on its structure. One is called as what? The purine, and another one is pyrimidine. So purine, this category containing two type of nitrogenous base. One is what? Adenine, and another one is guanine. Adenine and guanine, this can be represented with A and G. Always use the capital A and capital G to represent these two nitrogenous bases, adenine, guanine. Why? Because these are double cyclic ones. You see the structure here, adenine and purine. Here you can, this is what the purine category. Adenine and guanine, these are double cyclic. And such a type of double cyclic nitrogenous bases are called as what? The purine. And it belongs to adenine and guanine. Both are there in DNA and RNA. RNA and DNA, both are made with adenine and guanine only. Whereas come to the pyrimidine, it is monocyclic, only one cycle. It is simple in its construction. It consists of what? The cytosine and thymine. And one more, that is what? The uracil. Uracil is also there. It is also what? A unicyclic pyrimidine only. But why it is given in a circle? Because this uracil present only in RNA. It is not there in our DNA. The DNA consists of these two pyrimidine, cytosine and thymidine. The cytosine and thymine. Uracil is not there. Whereas in case of RNA, there is no thymine, only cytosine and uracil. So these three things, the adenine, guanine, and cytosine, these three things are common in both DNA and RNA. Whereas in case of DNA, thymine is there. Whereas in case of RNA, instead of thymine, uracil also here. So this is what the two type of the nitrogenous bases. One is purine. It includes what? The adenine and guanine, is double cyclic, and another one is pyrimidine, and it consists of only unicyclic nitrogenous bases, and cytosine, uracil, and thymine. Cytosine and uracil present only in RNA, and cytosine and thymine present in the, as I told you, purine is common, adenine and guanine. Cytosine is also common in what? The DNA and RNA, whereas thymine present only in the DNA, and RNA, instead of thymine, uracil only there. Right. The adenine, thymine, guanine, cytosine, these are the nitrogenous bases in DNA, and adenine, uracil, guanine, cytosine, these are the nitrogenous bases present in the RNA. Then same structure, adenine, guanine, double cyclic, right? Purine only. Adenine and guanine belongs to the category purine. It is double cyclic. Whereas these three nitrogenous bases, cytosine, uracil, and thymine, belongs to the unicyclic category called as what? The pyrimidine. And cytosine and thymine present only in the DNA, whereas uracil and cytosine present in the RNA. Now we are discussing in detail about the three different components of the nitrogen, this genetic material, both RNA and DNA. Right. Then come to the second one, that is pentose sugar. Pentose sugar is also there in both the DNA and RNA. And the backbone of this genetic material, nucleic acid, is made with these three components. This back bond consists of what? The nitrogenous bases, then pentose sugar and phosphate molecule. Then come to the pentose sugar in both DNA and RNA, having five carbon atoms. Here you can see the first carbon atom, second carbon atom, third carbon atom. That is what the fifth carbon atom. But here, this is what the pentose sugar present in the RNA. First carbon atom, second carbon atom, third, fourth, and this is what the fifth carbon atom. It is what the pentose sugar only in both RNA and DNA. But in DNA, pentose sugar is deoxyribose sugar, and in RNA, it is simple ribose sugar only. And what is the basic difference between these two pentose sugar? You see the position, second position of carbon atom in the pentose sugar. In case of what? Deoxyribose sugar, why it is called as deoxy? See, in the second position, second carbon atom, it is connected to what? This hydrogen only. This valency, it is 4. It is connected to what? This hydrogen atom only. Whereas here, in case of ribose sugar, instead of what you can say hydrogen, it is between these two type of pentose sugar. What is deoxyribose sugar? That is only in DNA, and because of that, it is stable, more stable than what you can say RNA. And in second position of what this pentose sugar in RNA, instead of hydrogen, you can find OH. See the color difference. Here it is given in pink color OH, and here it is in blue color. You can find this H only. This is what the basic difference between the pentose sugar. Another component present in the nucleic acid, both RNA and DNA. Then we will move to the phosphate group. It is common in both RNA and DNA. This is what the phosphate molecule connected to this pentose sugar. And let us see how these components connect each other in nucleic acids. There are three components. Remember always. One is what? The nitrogenous base. See the different type of nitrogenous bases present in RNA and DNA. In DNA, remember only the thymine, then cytosine, guanine, and adenine. Whereas in case of RNA, all the things will be there except the thymine. Instead of thymine, uracil will be there. And that only connected to the what you can say pentose sugar. In DNA, it is what? The ribose sugar only, deoxyribose sugar only. Whereas in case of RNA, that is what? The ribose sugar. Only difference in the second position of the carbon atom in the pentose sugar. And third one is what? The phosphate group, and it is common in both DNA and RNA. So let us see how it is bind together to get what? The structure of both RNA and DNA. So here, first one is what? The nucleosides, or we call it as what? The ribosides. Nucleosides or ribosides. And these nucleosides are produced with these two components. One is nitrogenous base and another one is pentose sugar. So thing is very simple, that pentose sugar is located exactly at the center. One side it is connected to the nitrogenous base, another side it is connected to the phosphate group. Whenever this pentose sugar connected to this nitrogenous base, get a nucleoside, or we call it as what? The ribosides only. So the first carbon atom of the pentose sugar, here you can find the first carbon atom. I told you the first carbon atom of the pentose sugar. First carbon atom of pentose sugar. In case of DNA as well as in case of RNA, connection will be in the same way only. And the first carbon atom of the pentose sugar only connected to the 9th carbon atom of purine. Purine means what? This is what? The purine, dibyl cyclic 1. 9th carbon atom of the purine connected to the first carbon atom of the pentose sugar. Whereas in case of what? The pyrimidine, it is unicyclic only. And the third carbon atom of the pyrimidine only connected to what? The first carbon atom of this pentose sugar. For getting what? The nucleoside, or we call it as what? The ribosides. Let us see different kinds of what? This nucleoside present in both RNA and DNA. These are the nucleosides. Here you can see the uridine. It is what the nucleoside in the RNA only. In nucleoside, only what the pentose sugar and nitrogenous base. Here, pentose sugar and nitrogenous base. These are what the purines and these are pyrimidine of what the RNA. And in DNA also, we can find the pyrimidine here connected to the first carbon atom of the pentose sugar and the third carbon atom of what this pyrimidine and ninth carbon atom of purine connected to the first carbon atom of the pentose sugar here. And these are the what you can say 4 different kinds of nucleoside present in RNA, and these are the 4 type of nucleoside present in the DNA. And the pentose sugar, remember, the pentose sugar present in the RNA is what normal ribose sugar only, whereas in case of DNA, see the second position, instead of OH, H is there, that is called as what deoxyribo, what you can say pentose sugar. And these are the nucleosides of this DNA. See, uridine, that means it is what nitrogenous base, and it is what the pentose sugar. Cytidine, it is what the cytosine and pentose sugar. Adenosine, it is adenine and pentose sugar. Then guanosine, guanine and pentose sugar. Guanine normally represented with the G only all the time. Then G connected to the pentose sugar here, and here it is what the ribose sugar only, because it is what the nucleosides of RNA, right? Then uridine, it is what U, and it is what the C, this is what A, and this one is what the G only, right? And these are the nucleoside. Nucleoside means what the combination of only nitrogenous base and the pentose sugar. Maybe it's what ribose sugar or deoxyribose sugar. In case of RNA, it is ribose sugar. In case of DNA, it is what the deoxyribose sugar only. And in case of DNA, we can find 4 type of nucleosides, 4 type of ribosides or nucleoside. One is deoxythymidine. Here it is deoxyribose sugar, and it is thymine, T. Then this is what the deoxycytidine, and here it is what deoxyribose sugar and C, and it is what deoxyadenosine. Pentose sugar is what deoxyribose sugar, and it is what adenine, and it is what deoxyguanosine, and here it is G, guanine. Ribose, nucleoside is a combination of only this pentose sugar and this what you can say nitrogenous base. It is different from RNA and DNA in structure. In RNA, all the pentose sugars are ribose sugar only, whereas in DNA, these are all deoxyribose sugar. And one more difference, in case of RNA, you can find U here. Uracil is there. In case of what the RNA, but in case of DNA, uracil is not there. Instead of, we can find this thymine here. That is what deoxythymidine, whereas deoxyuridine is not there. And what you can say that another one, that is thymine, is also not there in case of RNA, right? That you have to remember. Then come to the next level. Now we are constructing that this nucleoside. Nucleoside means it consists of what this pentose sugar and a nitrogenous base. And this nucleoside connected to the phosphate group in another part of what this pentose sugar. As I told you, this is pentose sugar. It is exactly at the center, and it is connected to the nitrogenous base with the help of the first carbon atom in one side, and fifth carbon atom of the pentose sugar only connected with the phosphate group in another side. Fifth carbon atom, this is what the first position, second position, third, fourth, and this is what the fifth carbon atom, and fifth carbon atom only connected to the phosphate molecule to get some more complex molecule here, and that only we call it as what the nucleotides. So nucleoside, then nucleotide, and this part is called as what the what you can say nucleoside. This is called as what the nucleoside, and whenever this nucleoside is connected to the phosphate molecule, we call it as what the nucleotides only. Right. It is a complex molecule consists of three components. All the three components of what nucleic acids are joined here together. And the first carbon atom of that this pentose sugar only connected to the nitrogenous base, and fifth carbon atom of the pentose sugar only connected to the phosphate molecule. In all cases, both in DNA and RNA. We will move to another one, nucleotides in DNA. Of nucleotides, all the nucleosides of the DNA connected to the phosphate molecule. So naturally, we get what the four type of nucleotides in DNA. Simple. And deoxyguanilic acid, that is DGMP, deoxyguanilic monophosphate, deoxyguanosine monophosphate, DGMP, guanosine monophosphate. This phosphate molecule connected to the fifth carbon atom here. It is what the guanine, guanosine, and it is what the deoxyribose sugar. Here we can find only H in the second position. So deoxyguanilic acid or deoxyguanosine monophosphate. And this is what the another nucleotide in DNA called as what deoxythymidilic acid or deoxythymine monophosphate. And it is thymine, and it is what the deoxy sugar, and it is what the phosphate molecule. And similarly, another nucleotide in the DNA is deoxyadenilic acid or deoxyadenosine monophosphate. And here nitrogenous base is what the adenine only, and it is connected to the first carbon atom of the pentose sugar, deoxyribose sugar only, and the fifth carbon atom connected to the phosphate molecule. That is what deoxyadenilic acid or deoxyadenine monophosphate. It's a monophosphate only. And next one, nucleotide present in the DNA. The pentose sugar, deoxyribose sugar only connected to the cytosine here with the first carbon atom and fifth carbon atom connected to the phosphate molecule. That is called as what deoxycytidilic acid or deoxycytosine monophosphate. Cytosine monophosphate. It is deoxy only. Deoxy nucleic acids. I mean, it is what the deoxycytidilic acid, deoxycytidine monophosphates. And it is what deoxyribose sugar, right? Then come to what these nucleotides in RNA. Nucleotides present in the RNA. Four types are there. And the nucleosides of the RNA only connected to the phosphate molecule. Here you can see this is what the ribose sugar. And this second position, you can find this OH group here. It is what the ribose sugar only connected to adenine here by using the first carbon atom and fifth carbon atom connected to the phosphate molecule to form what adenilic acid or AMP, adenosine monophosphate, adenilic acid or adenosine monophosphate. Why it is called as adenosine monophosphate? Because nitrogenous base is what adenine only, and here sugar is what the ribose sugar only. And guanilic acid and or guanosine monophosphate, and here guanine is there, and it is connected to the first carbon atom of the pentose sugar. Pentose sugar is what ribose sugar only. And third one, cytosine only connected here. And cytosine connected to what this ribose sugar by using the first carbon atom, and fifth carbon atom is connected to the phosphate molecule here, and it is called as what CMP. That is what cytidilic acid, CMP. And another one, this is only in RNA, not in the DNA, because uracil present only the RNA, not in DNA. And nitrogenous base is what the pyrimidine only here, uracil connected to the pentose sugar, ribose sugar only with the first carbon atom, and phosphate molecule connected with the fifth carbon atom to form another one that is called as what the uridylic acid or uracil monophosphates. These are the nucleotides. So nucleotide includes three components. All the three components are present in the nucleotide, and it consists of what the pentose sugar, also containing what nitrogenous base, and another one is what the phosphate molecule. And first carbon atom of the pentose sugar only connected to the nitrogenous base all the time in both DNA and RNA, and fifth carbon atom of the pentose sugar always connected to the phosphate molecule in both DNA and RNA. So it is developing like this only. Nucleosides. Nucleoside means only two components, nitrogenous base as well as what the pentose sugar. And whenever the nucleoside connected to the phosphate molecule by using the fifth carbon atom of the pentose sugar get little more complicated one, that is called as what the nucleotides. Whenever nucleotides connect each other, these nucleotides are connected each other with a phosphate molecule to get what a strand, and that only we call it as what the nucleic acids. So nucleic acid is nothing but the polymerized these nucleotides only. Whenever these nucleotides connect each other, we will see the connection of the nucleotide, how it is connect each other to get this strand nucleic acid. It may be single stranded or it may be double stranded, right? And this is what the structure of the nucleic acids, RNA. As I told you, RNA is single stranded. Maybe studied the structure of the RNA. It is single stranded and it consists of what? Only these four nitrogenous bases: cytosine, cytosine, then guanine, adenine, and uracil. No thymine in RNA. Instead of thymine, only uracil. Remember all the time, this nitrogenous base present in the RNA and nitrogenous bases present in the DNA, right? And this is what you can say. I told you about the polarity. It is what a long strand. See, this is what the what you can say pentose sugar, and here we can see cytosine, adenine, uracil, and cytosine in case of what the RNA. There is no thymine in case of what the RNA. So cytosine is there, adenine is there, uracil is there, and these are all connected to the first carbon atom of the pentose sugar in one side. And phosphate molecule, you know, it is connected to another end, and it is connected to the fifth carbon atom of the pentose sugar only. And this phosphate molecule connect each other. That fifth carbon atom of this pentose sugar connected to the third carbon atom with a phosphate molecule. And this phosphate molecule only connect this nucleotide. These are all nucleotides. This is what nucleotide, and this is also nucleotide. All these nucleotides connect each other with what the phosphate molecule by using the fifth carbon atom as well as what the third carbon atom, and to form a strand like this, a strand with a number of nucleotides, and that only we call it as what the nucleic acid strand, or in case of RNA, it is only a single strand. See, here you can see the strand with what you can say nitrogenous base CAUC, like we can find all these nucleotides connect each other with the phosphate molecule by using the third carbon atom as well as the fifth carbon atom to form a strand. It's having a polarity, having a definite end. See the strand of what you can say third position, carbon position of the pentose sugar is free, and that is why we call it as what the 3' end of this strand. And third one is connected to the phosphate molecule in the chain, but fifth carbon atom is free here, and that's why this end is called as what the 5' end. This is what the way to give polarity to a strand. It is a 3' end, 5' end. It is there in both the DNA and RNA. While discussing in the first slide, I told you DNA is what a double stranded. Double stranded. There are two strands here, and but having a definite polarity. This end is what the 5' end, and another one is what the 3' end. This is what the 5' and this one is what the 3'. And another one more strand is there in DNA, but it is running oppositely. If it is 5', this will be 3', and here it is 3', this will be 5', and it is what the anti-parallel. These are running parallelly but anti-parallel. One is from 5' to 3', another strand is from 3' to 5'. We will see how these kinds of arrangement is possible in DNA. While discussing about the structure of DNA, you will get more detail about this arrangement. So this is what the nucleic acids. See, nucleic acid is nothing but the long chain of nucleotides only, and all the nucleotides. This is one nucleotide. Like we can find thousands of nucleotide in RNA. These are all connected each other with the phosphate molecule by using what the third carbon atom of pentose sugar and fifth carbon atom of the pentose sugar. And one end of the strand, to form a strand, one end of the strand, the fifth carbon atom is free here. You can find that free carbon atom here, fifth one, and it is called as what, the 5' end, whereas another end, the third one is free, we call it as what, the 3' end. And see, leave these two and take all other nucleotide present in between these two, what you can say, terminal nucleotide. It is what, the last or the first nucleotide, the last one. And see all the other nucleotides in between. In between, you can say many nucleotides. In all the nucleotides, third one is connected to the phosphate, and the fifth one is also connected to the phosphate. They are all involved. Third carbon atom and the fifth carbon atoms are involved in the chain formation. Whereas this end and that end, or one is free. In one side, third carbon atom is free. We call it as 3' end, and another end, fifth one is free, and we call it as what, the 5' ends only. Right. Then this is what the nucleic acids DNA. Same way only it constructs the nucleotides. You can find a nucleotide here, here with the guanine. Here, guanine is there. Then pentose sugar is there. Phosphate molecule is there. And here thymine is there. For pentose sugar is there. And phosphate molecule is here. Here cytosine, pentose sugar, and that phosphate molecule. Here guanine, pentose sugar, then phosphate molecule. Thymine, pentose sugar, phosphate molecule. Like we can see cytosine, then pentose sugar and phosphate molecule to form one strand here. This strand is running from 5' to 3' end. And this end you see that the fifth carbon atom of the pentose sugar is free here. So we call it as what, the 5' end. This is what, the 5' end. And see the another end, fifth one is connected to the phosphate molecule and involved in this chain formation. But the third one is free, so we call it as what, the 3' end. So this strand is what this is one strand of the DNA, and it is 5' 3' direction only. And it is what, the double stranded. We can find another strand in the next side, but it is running opposite direction. That is from the 3' 5' direction only. And such a type of arrangement only keeping, you see, nitrogenous base of this strand here, it is in the center, it is directed towards the center, and nitrogenous base of this strand is also directed towards the center. So this arrangement and the parallel arrangement of these two strands only keeping the nitrogenous base exactly at the center. And here we can see the same arrangement. Here it is what, the pentose sugar, and it is what, the phosphate molecule. It is nitrogenous base, C, and here it is nitrogenous base, pentose sugar and phosphate molecule. And here G, it is what, nitrogenous base, and it is what, the pentose sugar, deoxyribose sugar only, phosphate molecule. So like that, another strand is also there, but it is running anti-parallelly. It is from 5' 3', and C, it is from the 3' to 5' direction only. And such a type of anti-parallel arrangement of these two strands only helping the nitrogenous base to keep exactly at the center. What is the need to keep this nitrogenous base at the center? And these two strands, phosphate molecules are towards the both side. Phosphate, here we can find the phosphate molecule, here we can find the phosphate molecule, only because of what, this anti-parallel arrangement, and nitrogenous bases are exactly at the center. And while discussing about the DNA, I told you it is complementary. These two strands are complementary. Because here you can find guanine, nitrogenous base called as what, guanine. And this guanine is what connected with the cytosine here. This is what a pair. Always pairing in DNA take place between the guanine and cytosine only. and guanine bind with cytosine with the help of hydrogen bond. So there are three facility guanine and cytosine. Whereas in case of thymine, you can find it's connect always with the adenine only. Guanine connected with thymine connected with the adenine only. But here only two facility for making the hydrogen bond. So always thymine connected to adenine with the two hydrogen bond, whereas cytosine connected to guanine with the three hydrogen bonds. And here cytosine connected to the guanine with the three hydrogen bonds. And here we can find the connection between adenine and thymine with the hydrogen bond, and only two facilities here, right? And here cytosine connected to the guanine with the three hydrogen bonds, right? So adenine here connected to the thymine with only two hydrogen bonds. And only because of what this anti-parallel arrangement. And it is complementary. Complementary means what? Always this guanine come close to the cytosine, and thymine come close to adenine. And adenine cannot connected with the guanine. Adenine cannot connected with the cytosine. Always connection will be there between the guanine and cytosine, and always what the adenine connected to the thymine only, and adenine can connected with the thymine with the two hydrogen bond, and the guanine connected with the cytosine with the three hydrogen bond. Normally we used to draw this connection between the guanine and cytosine with the dotted lines only, because it is very strength is very less, and this bond is what very weak. And thymine is, if you increase the temperature, naturally this bond will break and these two strands move apart each other. But whenever it is getting the opportunity, whenever it is getting the suitable condition, again it will come back and go for pairing, right? So that's what that is what the importance of DNA. RNA is not like, once it is disintegrated, it's not possible to get it back. Whereas DNA, whenever it is getting the opportunity, whenever it is getting the suitable condition, definitely they will come back and get what the original shape, right? So this is what the detailed structure of the nucleic acid called as what the DNA. As I told you, there are two strands here. This is what one strand, and this is what the another strand. There are two strands here. Nucleotide strands only connected each other with the phosphate molecule. See that first carbon atom, it's the third carbon atom and fifth carbon atom only involved for this connection to form a long chain of nucleotides. And having what direction here, the fifth carbon atom is free. Here you can find the free fifth carbon atom, and here it is third one, here fifth one is free 5' and here it is what the 3' only, right? And it is running anti-parallelly, like 5', 3', and another one is what the 3', 5' direction, anti-parallel. And this anti-parallel arrangement of the chain of the nucleotide only keeping the nitrogenous base exactly at the center and phosphate molecule towards both sides. And because of this arrangement only, it's possible for a bond between the thymine and adenine with the two hydrogen bond, daughter lines only, and guanine with some more stable structure. It is double stranded with, but you can say hydrogen bond is connect each other, get what some more stable structure. That's why I told you in the beginning, DNA is more stable than RNA, but at the same time, it can go for a slow chain. That is very essential for what you can say evolutionary purpose. We have to change the character, but the change of the character very, very slowly, only not very suddenly. We should not change our body character immediately due to what the changes happening in the physical factors of the environment or the physiological aspects of the individual, or it should not change according to the age of the individual. That should be stable. Then only we will able to express a character constantly in the body, right? Then this is what the base pairs. Come to the concept of base pairs. This is what the nucleotide chain. It is what another nucleotide only keeping this nitrogenous base exactly at the center and phosphate molecule towards both ends. See, adenine always come close to the thymine and produce what? Two hydrogen bond. Guanine come close to the cytosine and go for three hydrogen bond and getting a pair here. And that only we call it as what? Base pairs. Base means what? The nitrogenous base only. It is go for pairing with the hydrogen bond. But this pairing always between the adenine and thymine and cytosine and guanine only. And such a type of pair only we call it as what? The base pairs, and these are the complementary base pair. I told you it is complementary to each other. These two strands are complementary to each other, and these are the complementary pairs. Thymine always with what? Adenine only with two hydrogen bond, and guanine always connected to the cytosine with the three hydrogen bond, and these are the complementary base pairs. Normally, you can find such type of symbol in the textbook. BP, that is base pair only, not blood pressure. It is base pair. Always pairing take place between adenine and thymine, HE, and cytosine and guanine, that is CG. Remember this pair, right? See, this is what the detailed structure of what the DNA strand. As I told you, DNA, it's double-stranded, and see, this is what one strand. We can find this purine nitrogenous base here, and it is connected to the pentose sugar, and fifth carbon atom is connected to the phosphate. And fifth carbon atom is connected to phosphate, and third one is also connected to the phosphate. So you just take it as a strand, and here it is what? The 5' N, and the third position of this pentose sugar is free here, and we call it as what? The 3' strand. Whereas this one, the fifth position is free, we call it as 5' end of DNA, and here third position of the pentose sugar is free, so we call it as what? The 3' N. And this arrangement is anti-parallel, so it keeps the nitrogenous base exactly at the center, and always these base complementary bases only come together and making this bond, and it is called as what? The base pair. So you see that deoxyguanosine, the DG, that is what? Deoxy only, that is what? The nucleotide in the DNA, and the guanine are connected to cytosine with the three hydrogen bond, and here we can see the connection between the guanine and cytosine with the three hydrogen bond, whereas adenine connected to thymine only with the two hydrogen bond. Remember always, but a stable structure to this DNA, and it can be broken. You know, it is what? Very weak bond, only hydrogen bond. That is why it is given in dotted line here, and it is faded also. It's not so strong. So by heating, we can break it. You may be studied such type of experiment in what you can say in some other chapters, right? Breaking it, but whenever it is getting what? The condition back, naturally this will go for pairing, because these can attract each other. They go for pairing, and at least in the right hand direction, and get its what? This stable structure. So these two coils, you know, it is, I mean, these two chains, anti-parallel chains, go for coiling. Go for coiling in a right-handed position. Go for coiling in right-hand position to get more stable functional structure. And this is what the always you may be seeing the structure of this DNA. It will be like this only, right? You may be notice such type of things will be there, and it is also connected each other with the hydrogen bond, because these two strands, anti-parallel strands, go for coiling in right-handed position and get what? Somewhat stable structure, and this is what the functional structure of DNA present in all the individual, and this DNA only working as a genetic material in most of the individual, except what? Retroviruses. Because in retroviruses, DNA is not there. They have only RNA, single stranded, and it that only working in their body as a genetic material to express the character that only pass to the next generation. Disadvantage is that it can go for mutation so quickly because it is single stranded. Right. Once it is changed its chemical nature, naturally it expresses totally different character. That is why in case of our retrovirus, it's very difficult to find out the medicine. Whenever we invent a medicine against that particular retrovirus, because in future what will happen, it suddenly changes character and appears in different way, and we may not able to treat the disease with what they invented medicines, right? Because it keep on changing its what the chemical nature to the process factor of the environment can make a lot of changes in their genetic material, right? So we will move to another slide, right? See the double-stranded structure of DNA after coiling. It is what the sugar phosphate backbone only. And this is what one strand running from 5' direction to 3' direction here. And another strand, it's running from 3' direction to what the 5' direction here. And we can see the coiling of the DNA. And this is called as what? One pitch. One pitch. Pitch of what? The DNA. One pitch of DNA. One pitch, one coil of DNA is what? One pitch. And the length of what this one pitch of the DNA is what? 3.4 nanometer. 4 nanometer. This is what the pitch length. Pitch length. Pitch means what? One coil of the DNA is called as what? The pitch, and length is what? 3.4 nanometer. That is what the pitch length. And the distance between, in one pitch, we can find 10 nitrogenous bases. 10, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. It's not there in the picture, but 10 nitrogenous bases are there in one pitch. So naturally, the distance between two nitrogenous base in the DNA is only 0.34 nanometer. That is what the distance between two nitrogenous base in the double-stranded DNA. That is 0.34 nanometer. And this is what the pitch, one coil of the DNA, always it go for coiling in right-hand position. And getting many coil in one coil consists of what? The 10 nitrogenous base, and length is only 3.4 nanometer. That is what we call it as what? The pitch length. And see the distance is only 2 nanometer between these two strands. Width of the DNA is 2 nanometer, and like many pitch will be there in the DNA, depend on the chemical nature, number of nucleotide present, number of pitch will be there, but every pitch consists of only 10 nitrogenous base pair, and the length is only 3.4 nanometer, and the distance between two nitrogenous base pair in the DNA is always 0.34 nanometer, because 10 nitrogenous base is 3.4 nanometer, 0.34 nanometer only. And this is what the phosphorus molecule, whenever it go for coiling, you get a structure like this. Carbon sugar phosphate backbones are there, and hydrogen bonds are also there to connect one strand to another strand, right, to form these bases. Then come to the salient features of DNA. We have discussed mostly all the features while discussing about the structure of the DNA. So all of you know it is made with the two nucleotide chains. Now we are discussing in detail about the salient features of the DNA only, not RNA. It is made with the two nucleotide chains. And backbone of the DNA is with pentose sugar, that is deoxyribose sugar only, then phosphate and nitrogenous base. And this is what the backbone of the DNA. Phosphate molecule, pentose sugar, and nitrogenous bases. Then phosphate projected outside. See, all the phosphate molecules are projected to outside only. And nitrogenous bases to inside. See, nitrogenous bases are towards the inside only. And these two chains are having anti-parallel polarity. See, anti-parallel polarity. And here, third one is free. Third carbon atom, 1, 2, 3. Third carbon atom is free. And here, what the fifth carbon atom, 1, 2, 3, 4, 5. It is what the anti-polarity. The two polarity, that is 3 prime, 5 prime, and another one is running from 5 prime to 3 prime direction only. Or 5 prime means 5 dash, or 3 prime means 3 dash. So two chain pairs with a hydrogen bond from base pairs. So these are the bases, nitrogenous bases of these two strands. These two strands always go for binding with a hydrogen bond to form the base pairs. See, cytosine bind with the guanine with the 3 hydrogen bond to form the base pair, and adenine with the thymine connect each other with 2 hydrogen bond. So to form these complementary base pairs only, adenine always with the thymine and cytosine always with the guanine, and form what the complementary base pairs, C double bond G and adenine, C triple bond is a mistake here, triple bond G and adenine double bond T, adenine double bond T, cytosine triple bond G, right? In base pair, one is purine and another one is pyrimidine. You see the base pair. In every base pair, adenine is purine, thymine is pyrimidine. And here guanine is purine, cytosine is pyrimidine. Purine means what? Having the double cyclic structure, and pyrimidine is what? Single cyclic structure. So always this bond formation take place between the purine and pyrimidine only. So base pairs, one is purine and another one is pyrimidine only. See, adenine is purine, thymine is pyrimidine. Then guanine is purine and cytosine is pyrimidine. And this chain coil in a right hand position. I told you, after the formation of this, double stranded structure, go for coiling it in right hand position. And one pitch length is 3.4 nanometer. Pitch means what? One coil. In DNA is called as what? The pitch. And length is what? 3.4 nanometer. One pitch consists of 10 base pairs. So the distance between two base pair is only 0.34 nanometer. And see the total number of purine in a DNA. You can take any DNA, any length, but the total number of purine is always equal to the total number of pyrimidine. Total, because always bearing take place between the purine and pyrimidine. So naturally, you take the DNA, total number of purine is always equal to the total number of pyrimidine. And see, the total number of adenine is also equal to the total number of thymine, and total number of guanine is equal to the total number of cytosine only. So total number of purine always equal to total number of pyrimidine in DNA. That is not there in RNA because it is unistandard. And DNA is more stable than RNA. And see here, the number of adenine is not equal to guanine in DNA. Number of thymine is not equal to guanine or cytosine. And number of adenine is always equal to number of thymine in DNA, and number of cytosine is always equal to total number of guanine present in the DNA. But the guanine is not equal to adenine or not equal to thymine. Cytosine is not equal to thymine or adenine. Adenine is not equal to guanine and cytosine, and thymine is also not equal to cytosine and guanine. But always the number will be same, and C and G number also will be same, because they between these two only the pair will take place, always between the purine and pyrimidine. So total number of purine in a DNA is always equal to the total number of pyrimidine, right? Then DNA is more stable than RNA, because as I told you, RNA is what we will see later in very detail about what the reason for the stability of the DNA, right? And RNA is not very stable because it is single stranded, having so many other disadvantages. Of course, in the nature, RNA only produced as a genetic material initially, but because of the disadvantage, it's what the process of evolution, changes are happening. Always what the changes are in the progressive direction only. So naturally, a lot of modifications comes into the genetic material, and finally it created the DNA as a genetic material, having a lot of advantages. So now only the DNA, selected by the nature. Now still we can find the DNA as a genetic material in all the individual except for the retroviruses, right? Then the total number of G is equal to the total number of Z, and total number of A is equal to the total number of T. So that is these are all what salient features of the DNA already we discussed while constructing the structure. And this is what the double stranded structure is. Go for recoiling in the right hand position, and length of the DNA varies, and the number of nucleotide present in the DNA varies, but it is more stable than what the RNA, acting in the body of an individual as a genetic material, and this DNA only express a character in the individual in the form of what the proteins. That we will see later. You may be heard about the protein synthesis. And protein synthesis involved two processes mainly. One is what the transcription and another one is translation. From this DNA, first that RNA will be transcribed, because all the messages related to the chemical nature of the protein should be what you can say should be given in the form of message in the machinery for protein synthesis. That is why the entire messages present in the DNA is copied to RNA, because DNA cannot go for protein synthesis, cannot involve directly in the protein synthesis, because they are storing the information there. So whenever you are getting a certificate, you are submitting the certificate, not the original, the duplicate one only, because original you are keeping always for the reference. Right. Similarly, DNA is keeping all the information about the type of protein produced in the cell. And this information, they are simply copying to the RNA. That process only we call it as what the transcription. Then this copied information in the RNA only taken to the machinery in the cytoplasm, where it go for that process of translation, the real process to produce the protein. So actually the information regarding the protein, chemical nature of the protein is given by the DNA only. That is why the DNA is considered as what the genetic material. And RNA is what a messenger. It is giving a message, and it is just to copy the information from the DNA to prepare a particular protein with a definite chemical nature. That only determine the character of the individual. For example, the color of the body. Color of the body is a character, and it is due to what preparation of a protein. We call it as what the melanin. Melanin pigments only determine the color of the individual, and it is a protein actually produced by the genetic material DNA present in the body. Right. Then we will see some details about the difference between the DNA and RNA. Most of the differences are known to all of you, right? DNA and RNA. See, DNA is mainly occurs in nucleus. You maybe heard about the chromosome. We have 23 pairs of chromosome in our nucleus. And this chromosome consists of mainly the DNA. Of course, it also containing some proteins like a histone and non-histone protein, right? And but the content present in the chromosome to express a character is what the DNA only. And it is present only in the nucleus, and it is well protected in with what the nuclear envelope, right? And whereas in case of RNA, there is no RNA in the nucleus. Of course, RNA is synthesized in the nucleus with the help of what the DNA only, but in the nucleolar region. You may be heard about the nucleolus. It is what dark dense area in the nucleus. This site is exclusively for synthesizing this RNA only. That is why it looks very dark in color. That nucleolus inside the nucleus, that area looks very dark with the denser, what you can say, nucleoplasm, only because of the RNA. But immediately after producing the RNA in the nucleus, this will be taken directly to the cytoplasm. So we will able to see this RNA mainly in the cytoplasm only, and DNA we will not able to see in the cytoplasm. Of course, DNA is there in the cytoplasm, not in the cytoplasm, but in mitochondria it is there. And in case of plant, chloroplast, you may be heard about the mitochondria and chloroplast. These two are called as what the autonomous cell organelle, because they are having the genetic material inside. They are able to prepare the enzymes for their function by using that circular DNA present in both cell organelle, that mitochondria and chloroplast. See, it is a genetic material. DNA is a genetic material, and RNA is not a genetic material, and it is only to translate the protein. But in retrovirus, it is acting as what the genetic material, and it is double-stranded. This RNA is single-stranded, and it is has a regular coiling DNA. It is antiparallel and go for coiling in right hand position, and such a type of coiling is not there in RNA, but irregularly. For example, in case of tRNA and rRNA, we can find some kinds of coiling to make it what a three-dimensional structure to perform a particular function. And so there is no such a regular coiling in RNA, and it consists of deoxyribose sugar. This is what the ribose sugar only. And nitrogenous bases are adenine, thymine, cytosine, and guanine. Whereas in case of RNA, what are the nitrogenous bases in RNA? This adenine is there, cytosine is there, guanine is there. These are common for both the DNA and RNA. Whereas in RNA, instead of thymine, uracil is there. Then hydrogen bond between base pairs in DNA, because while discussing about the structure, I told you nitrogenous base projected towards the center and making the bond with the hydrogen bond, and that is not there in case of the RNA, because it is single stranded only. The purine and pyrimidine base are equal in DNA, because always purine only connected to the pyrimidine, so naturally number will be equal. Whereas in case of RNA, as a single stranded one, the number is not same. It's very, sometimes purine will be more or pyrimidine will be more, depend on what the chemical nature of the RNA. And it can replicate, it can make its copy. DNA can be what you can say multiply through DNA replication, but this RNA cannot replicate. And DNA transcribes genetic information into RNA. That is called as what transcription. I told you DNA genetic information is there that can be copied to the RNA. That is called as what the transcription. But in case of RNA, no transcription, only translation. What is translation? The information copied directly from the DNA can be used for making what the protein. That is what the translation. And another one, it is only two types, nuclear and extranuclear. But the DNA present inside the nucleus or inside the nuclear envelope that is there in the chromosomes. In a cell, extranuclear means what? These, as I told you, that what you can say plastids, or in case of mitochondria, these two are autonomous cell organelle having a circular DNA in their what you can say cell organelle. That will be making what these two cell organelle autonomous, because they are able to produce what the enzyme, not at all depending what the nucleus for getting the enzyme to perform the cellular respiration in mitochondria or photosynthesis in chloroplast. They are self-sufficient. We call it as what the autonomous only, and having two type of what the DNA. In the nucleus it is there. Extranuclear means in the autonomous cell organelle. Whereas in case of what the RNA, it is there, three types are there, not two types, three types are there. mRNA, that is messenger RNA, then tRNA is transfer RNA, and rRNA, it is ribosomal RNA. We will see the structure of all these three different type of RNA while discussing about the process of translation and transcription. And DNA content in the cell of an individual is constant. In case of human being, as I told you, only 23 pairs of chromosome in a diploid cell and 23 chromosomes only in the haploid cell, and it is constant all the time. And it varies from cell to cell in an individual. You take an individual, cell to cell, we can find the differences. Some cells are very active. They are involving in the protein synthesis continuously, and some cells are very inactive. They are not producing much enzymes in their body because they don't want much enzyme to perform the work inside. So depend on the activity of the cell, the number of cell or the chemical nature of the RNA present in the cell varies. So individual to individual difference will be there. Even in the same individual, cell to cell, we can find lot of differences in the what you can say the total content of RNA present in the cell. So these are the main differences between the DNA and RNA. So once this question asked in the board examination, you find out some differences by heart it. Whenever you discuss in detail about, or whenever you study the structure of both the DNA and RNA, you can find lot of differences in their chemical nature, then functions and structure, etc. That only you have to put it into this table column and write the differences, DNA and RNA. Right? We will move to what you can say. That's all about what the structural detail of DNA and RNA. Then we will see some topic mentioned in the textbook. Nucleic acid is the real genetic material, not the protein. There was a big controversy between the scientists, which is the real genetic material. Is it a protein or is it a nucleic acids? See, nucleic acids, both DNA and RNA can duplicate, and but protein cannot duplicate. It cannot make the duplication. Means what? Making the exact copy without changing its chemical nature. And protein cannot be duplicated, whereas the DNA and RNA can be duplicated. So RNA and DNAs are called as what? The nucleic acid. So genetic material should multiply through the process of duplication before every cell division. So remember, whenever cell divide, for example, in case of human being, 23 pairs of chromosome present in a diploid cell. Whenever this diploid cell divided into two mitotically and produce two cells, and both the cells are also having what? 23 pairs of chromosome. Because before the cell division, naturally the entire genetic material or the chromosome present in the cell get duplicated. Then only it is possible to divide evenly. That is why we call such type of division is what? The equational division. So it is multiplying the genetic material, or it is increasing the genetic material through the process of what? Duplication only. So genetic material should multiply, or it should multiply through the duplication without changing. We are making the duplicate. Duplicate means what? We are taking Xerox copy, no change in their what you can say content. So such type of duplicates only we are making here before the cell division to divide the cell evenly into two, right? And only this nucleic acid can go for duplication, not protein. So protein cannot act as what? The genetic material. And nucleic acids are more stable than protein. Protein can degenerate. It can be degenerated depending on the climatic condition, right? And the genetic material should be stable and not degenerate to express a character. Because nucleic acid only expressing a character. If it is changing the chemical nature according to the climatic condition, naturally lot of defect will be there in the individual to express a character. Generation to generation, we can find a lot of difference in the character. And even in the individual, according to the climatic condition, we can find lot of difference in their expression of the character. So the nucleic acid should not, the genetic material should not change according to the physical standard or climatic condition or other aspects of the individual. It should be stable. So this nucleic acid is stable than protein. So nucleic acid only can act as a genetic material. And here we are discussing why nucleic acid is acting as a genetic material, not a protein. Because nucleic acid, can duplicate. Nucleic acids are more stable than protein, and DNA and RNA are able to mutate, but protein cannot. But mutation, it is a slow changes happening in the chemical nature of this genetic material only. It should be very slow, not very sudden. Then protein cannot mutate, but RNA can mutate or DNA can also mutate. So genetic material should change its chemical nature according to the change of the environment, but very slowly only through the process of mutation. As I told you, what happened if it is changing its chemical nature so quickly? That will create a lot of trouble. But it should provide the scope for a slow change. Then only we will able to make variations. We will able to make a new character in the coming generation, but these changes should be very, very slow. Then only our body will able to cope up with the changing environment. If you want to, for example, you just see the body character of the human being who lived around what you can say 10,000 or 20,000 years back, you may be learned in what you can say evolution chapter, and see the body character of the present generation, you can find lot of differences, and these differences should be there. It is very essential to cope our body with this environment. So environment in the past, before what you can say 10,000 or 20,000 years back was totally different. So if we want to cope our body with the changing environment, we should change our body character, but it should be very, very slow. If we are not changing, naturally we will not able to succeed in our life. We will not able to survive in the nation. You may be heard about the extinction of many individual dinosaurs. Once they ruled the entire world, but now it is totally extinct. It's just like now human being is ruling everywhere. Similarly, that dinosaurs, that giant reptiles only ruled everywhere. But now it is extinct forever. Why? Because they were not able to change their body character to cope up with the environment. Right. So it should be there. Genetic material should change the character. I mean, change the chemical nature through the process of mutation, but should be very, very slow and very gradual. The nucleic acid can store information. Information for what? Information for expressing a character by producing a particular protein. So maybe learn about the translation, how these informations are given in the DNA molecule. Is a sequence of nitrogenous bases in the DNA molecule. That is what the informations regarding what type of protein synthesized in the body. That information can easily store in the genetic material, but in protein it is not possible to store the informations. So because of this reason, nucleic acid can only act as a genetic material, not protein. There are 4 points mentioned here, and these two characters should be there in a nucleic acid. Nucleic acid should duplicate. Nucleic acid should be very stable, and nucleic acid should change its what the chemical nature very gradually, and nucleic acid should store the information regarding the expression of a character, and this can be satisfied only with what you can say, I mean, nucleic acid only. These are the characteristic features of the genetic material, not nucleic acid. That genetic material should, what you can say, duplicate. Genetic material should be stable, and nucleic acid should be go for mutation very slowly. And nucleic acid, genetic material should store the information. These characters can be satisfied only in the nucleic acid, not the protein. That is why nucleic acid is considered as what the genetic material, not protein. Protein cannot act in our body as a genetic material, but lot of controversies were there initially regarding the chemical nature of the genetic material. Is it a nucleic acid or it's a what, a proteins? So there are two what you can say nucleic acids, DNA and RNA. Of course, nucleic acid only can act as a genetic material because it can store information, can go for duplication, it go for mutation. But which nucleic acid is a good, what you can say, good for keeping the information? Is it DNA or is it RNA? We will see some experiments later to prove that the DNA is what the real genetic material. Because DNA can only act better than what you can say RNA as a genetic material. So DNA is the real genetic material, not RNA. In case of what the retrovirus, as I told you, there is no DNA. So naturally, the RNA only acting there in their body, retrovirus like AIDS virus as a genetic material. And RNA is the first evolved genetic material, no doubt, in the living organism. Still, it is acting as a genetic material for some viruses also, like what you can say retroviruses. And RNA has undergo a lot of chemical changes during the process of evolution and developed more stable chemical called DNA, and that only acting in most of the individual as what the genetic material. RNA only evolved initially as a genetic material in the living organism. See, every molecule in is undergoing change, having better character than RNA to store the information. That is why nature select DNA as a retrovirus as a genetic material, whereas all other individual except this retrovirus, it is replaced, and now DNA only acting, so it is not resistant to the sudden changes. It is not resistant because it is single stranded. If it is changing what the chemical nature, it cannot be repaired, or it is not possible to resist. Whenever it is getting what the climatic suitable condition back, again it go for making the double strand and act as what a functional genetic material, and it is repairable. Once it is broken, again it can be repaired, resist the sudden changes and also repair at the same time. It can also but it can say probable and slow changes that should be there in case of genetic material. That is why this most of the individual except this retrovirus acting as what the DNA only acting as what the genetic material. Then why DNA is more stable than RNA? This question asked once in the board examination. Why DNA is more stable than RNA? You know very well the detailed structure is showing more stability than this RNA. DNA regain its structure and character when it get appropriate condition. I told you DNA is double-stranded and connect each other with a hydrogen bond only. It can be broken because it is what a weak bond. But after breaking, whenever it is getting what an appropriate condition back, again, we go for making double-stranded functional structure, right? And OH group in RNA, OH group, where OH group is there in the RNA, the of the pentose sugar. Remember the position of the pentose sugar. Their OH group is there, and it is there only in RNA. But in case of DNA, it is H only, and it is very reactive and easily degradable. That is why the DNA is more stable than RNA. Then another one, this RNA can act as a catalyst. So unstable. Catalyst means what? The biological catalyst only. Catalyst means what? The chemical working in a chemical reaction in cellular level, but it is not directly involved. But its presence is very much necessary. And RNA can act as a catalyst, but not DNA. Then thymine in DNA provide more stability. Thymine is not there in RNA. Instead of thymine, RNA is having what? The uracil only. So that thymine present in the DNA can provide more stability. RNA mutate so fast, right, due to the single stranded nature only. That's why I told you that RNA viruses, normally go for mutation with faster rate, and the DNA is double stranded, so it is stable. Of course, it is undergoing the mutation, but very, very gradually. But it is essential for the evolution. Then, for the storage of genetic information, DNA is better, but its expression required RNA. So normally DNA only a good genetic material, or DNA only having that facility to store the information to express a character in the form of a protein. But DNA always required RNA to express the character. Remember the transcription and translation process. DNA is not able to express a character directly because it is only for the storage of information, always inside the nucleus only, always storing all kinds of information related to the expression of character by producing a protein. But always DNA express a character with the support of what? This RNA only. RNA is very much necessary for the expression of characters. Right. Then we will move to another slide. Packing of DNA helix. See, the DNA is very lengthy. See, in case of what the mammalian cell, the total length of the DNA comes around 2.2 meters. Because very lengthy, double-stranded, very lengthy one. And the cell itself is microscopic, and you see the size of the nucleus. How it is possible to pack this much length of the DNA in the nucleus? It should go for repeated coiling, and it should go for the repeated condensation. Such type of coiled condensed DNA only able to keep it in the what you can say, the nucleus. Whenever it go for coiling, so whenever we get the thread, we used to make the coils, so that we can uncoil and we can use it. So coiling should be very pakka, because there should be any difficulty in uncoiling. So whenever we want to express a character, we have to uncoil this DNA, then we have to express a character. So coiling should be very pakka, should be very systematic. It should be uncoiled at the time of what the expression of a character. So through this way only, this coiling and condensation, repeated coiling and condensation, only helping us to keep this much length of the DNA in a small microscopic nucleus. That only we call it as what, the packing of DNA helix. The total length of the DNA helix is calculated approximately by multiplying the total number of nitrogenous bases and distance between these two pairs. We already discussed the pitch and pitch length are distance between the nitrogenous base. So if we get the total number of nitrogenous base in DNA, we will able to calculate the total length, nanometer. Then it comes out to approximately 2.2 meter in a mammalian cell, including human being. It is far greater than the dimension of a typical nucleus. Nucleus is very microscopic and very microscopic, and cell itself, it's very microscopic, and nucleus is what inside as a small, what you can say, a dot. Is it possible to keep that much length of the DNA into that small microscopic nucleus? How is such a long polymer packed in the cell or the nucleus of the cell? That only we are going to discuss here now, packing of DNA helix, right? And see, if the length of E. coli DNA is, okay, this you can discuss with your teachers. So first we will discuss about the structure, then we will come back and we will discuss these problems. So DNA in prokaryote. Prokaryote means what? Bacteria. What is the problem of the prokaryote? Because it is not having the nuclear envelope, so that the genetic material is naked. It is there in the cytoplasm, not having any, what you can say, envelope. There is no boundary for protecting the genetic material in case of what, the prokaryotes like a bacteria. You can find this bacterial DNA. Even plasmid is also there in the cell cytoplasm only. Plasmid is what, an extra chromosomal DNA only, a circular both. This, what you can say, chromosomal DNA is circular and very lengthy, and this plasmid is also circular DNA only. So like E. coli does not have a well-defined nucleus. DNA is not scattered throughout the cell cytoplasm. Doesn't mean that no nucleus, so it is spread everywhere in the cell cytoplasm. Not like that. It is concentrated in a particular area, this DNA, and this DNA is negatively charged chemical only, and which is held with some positively charged protein in the region of the cell cytoplasm, and that only we call it as what, the nucleoid or the nuclear region. This is what nucleoid or the nuclear region. Of course, there is no boundary, but all these what negatively charged double-stranded DNA held with some positively charged protein present only in this region, not everywhere. And this region where this double-stranded DNA is located and it's connected and attached with the positively charged protein like histone or non-histone protein, and that area of the bacteria is called as what, the nucleoid or the nuclear region. In case of bacteria, there is no nucleus, only the nucleoid, only the nuclear region. This is what the region in which what the genetic material is kept, and this genetic material DNA is basically negative in charge, so naturally it attached with the positively charged protein present in that particular area. And this area looks what, very dark, dense in color, And the DNA in nucleoid is organized in large loop held by proteins. I told you protein is what positive in charge, and this DNA is going for repeated coiling in a particular way, and it's held with what the positively charged protein. See, this is what the what you can say double-stranded DNA go for coiling and connected to the positively charged protein. And such a type of very coiled DNA only located in the nucleoid region or the nuclear region of the prokaryote. Right. It's very coiled. You see length of the total length of the DNA, and such a very lengthy DNA is keeping in a small area inside the prokaryotic cell only because of what such a systematic coiling of the DNA only. See, in E. coli, 1.2 microns long, this DNA, having a circular genome consists of 2 million base pairs. In DNA of this E. coli consists of 2 million base pairs, and it is 1,000 times longer than the bacteria itself. And it is possible to keep this large DNA in the bacterial cytoplasm only through this supercoiling by binding it with the positively charged proteins. I told you in nuclear region, we can find the positively charged protein, and this positively charged protein only bind with the negatively charged protein, and negatively charged DNA is going for a supercoiling. Otherwise, it is not possible to keep inside the what you can say cell cytoplasm, because it's what 100 times, 1,000 times longer than the bacteria, and the total number of what these base pair present in the E. coli DNA is what 2 million. So it's go for supercoiling, and it is keeping in particular area of this bacteria. Then come to this DNA coiling in eukaryotes. We all belongs to the eukaryotes. Other than bacteria, all other individuals are called as what the eukaryotes only. Then come to the DNA coiling in eukaryotes. Say DNA in eukaryote is same negative in charge, which coiled around the positively charged protein called as what the histone proteins. Histone. I told you the protein present in the chromosome, histone and non-histone. The histone protein is large in its quantity, whereas non-histone protein is only for keeping the structure of the chromosome, and the content is very less. And this histone protein is positive in charge, and DNA is negative in charge. So the basic protein acquires positive charge due to large amino acid residue with a charged side chain, particularly the residue of lysine and residue of this arginine. This question asked in this first term examination, which are the amino acid present in the what you can say this positively charged histone protein, and the residue of lysine and residue of the arginine. These two are amino acid, and these amino acids only giving the positive charge to these proteins. This basic protein acquires a positive charge due to what the large amino acids residue, and these are the amino acid provide the protein charge to this basic protein, histone protein. One is lysine and another one is arginine. And this question only asked in the first time examination. Go through the questions and find out the answers. And histone proteins, are organized to form a unit of 8 molecules. That only we call it as what the histone octamer. You can see the structure. This is what the histone protein. There are 8 units. 1, 2, 3, 4, 5, 6, 7, 8 units. And 2 H2A proteins, H2A unit, and 2 H2B unit, and 2 H3 unit, and 2 H units. So this is what histones are organized to form a unit of 8 molecules. We call it as what the histone octamer only. And octamer, 8s are there. There are 2 H2A, there are 2 H2B, there are 2 H3, and there are 2 H. Total 8 molecules are there in a histone protein, and we call it as what collectively histone octamer. And this negatively charged DNA is wrapped around this positively charged histone octamer to form a structure called as what the nucleosome. Now we are discussing how this DNA is going for coiling in eukaryotic cell. This is very essential to keep it in a small space in the nucleus. Negatively charged DNA is going for coiling around the positively charged histone octamer. Eight molecules are there in the octamer to form a structure that is called as what the nucleosome. And each histone octamer is composed of two copies of each of the histone protein, H2A 2 times, H2B 2 times, H3 2 times, and H 2 times. Total 8 molecules in an octamer. That we call it as what the histone octamer. And these are the histone proteins given here in dark color, that round structure, two histone H2A molecule and two H2B molecule and H3 2 and H2, and this is what the histone octamer, histone protein, histone octamer. And the DNA, the red color it is given, that is wrapped around this histone octamer 2 times. And this is what the core of the histone molecule. And this DNA molecule, it is negatively charged, and histone is what positive in charge. So it wrapped around 2 times to form a unit called as what the nucleosomes. Histone protein is positive in charge, and the DNA is basically negative in charge. So negatively charged DNA is wrapped around the histone octamer 2 times, like that. It is moving like this 2 times and form what? A single unit. We call it as what? The nucleosome. Now we are discussing about the coiling of the DNA, how it is going for the systematic coiling to keep such a lengthy DNA into the nucleus. Then come to the details of the nucleosome. A typical nucleosome. Remember the structure of the nucleosome, 8 octamer. Molecules are there in a nucleosome, and that DNA is also wrapped around 2 times. So that is what the nucleosome, and it contains 200 base pairs of DNA helix. 2 times it's wrapped around. And it consists of what? The 200 base pairs in one nucleosome. And this nucleosome constitutes the repeating unit of structure in the nucleus called the chromatin. And this nucleosome again go for what you can say coiling to form another structure, because it is super coiled. Chromosome is what? It is what? A super coiled DNA only. DNA go for coiling to form the nucleosome. Again, this nucleosome constitute repeated coiling to form what? This chromatin. And this coiled long chain of what? This nucleosome connect each other with a linked DNA. Here we can see the linked DNA. This is what one nucleosome, and it is what another nucleosome, and the DNA is what you can say wrapped around this nucleosome, and it is coming, and it is what another coiling here, and another nucleosome, and one nucleosome connected with another nucleosome with what? A DNA here, and it is called as what? The linker DNA. So that only mentioned here, the coiling along the chain nucleosome connect each other with a linked DNA only. And nucleosome in a chromatins are seen as a beads of string. Beads of a string. Structure when is viewed under electron microscope. Whenever we are viewing this, what? The chromatin under electron microscope, we will see what? The what you can say nucleosome like this. Just like what? Number of beads in a mala. We can find such type of what? Number of beads. Just like what? A mala only, we can find arrangement of one nucleosome, another nucleosome connected with what? This linked DNA in a chromatin. Chromatin unit is what? Somewhat coiled nucleosomes only after connecting. And beads of string-like structure in chromatin is packed to form a chromatin fibers. Chromatin fibers. And see, first a nucleosome, then go for coiling to get the chromatin, then chromatin again go for repeated coiling, form a very long bead-shaped structure called as what? The chromatin fibers. And these chromatin fibers again go for coiling. and form what the final product, and it is called as what the chromosomes. We have 23 pairs of chromosome, and you take one chromosome and see how it is made with this DNA mainly only through what the repeated coiling around the histone proteins only. So the beads of string-like structure in chromatin is packed to form the chromatin fiber that are further coiled and condensed during the cell division, and it appears only during the cell division, particularly in the metaphase stage, and that only we call it as what the chromosome will appear only just before the cell division, right? And it is what a supercoiled one. In class 11 you may be learned that cell division, during cell division what is happening, to separate the chromosome into two, chromatin thread should be converted into what the chromosome through this repeated coiling. The same way only it coiled. First it produced what the nucleosome, then chromatin fiber, then chromatin fibers again undergo the repeated coiling and condensation, and it finally produce what this chromosome. So what is chromosome? Nothing but a super coiled, what you can say, DNA only. It's coiled with what histone protein, and for the stability, we can find some non-histone protein also. So the chromosome consists of only three things. The main thing is what the DNA. Another one is what the histone protein, and we can find little quantity of what the non-histone only. And we consider the chromosome as what the genetic material. Why? Because in chromosome, DNA only helping for the expression of character, and it should be stable, and it should be definite in an individual. Every individual, there is a particular number of chromosomes, right? If it is changed, naturally it affects very badly the life of the individual. It is not possible for a triploid condition in human being, or a haploid condition. Of course, the gametes are haploid, but what you can say, the cell should be diploid, not triploid or tetraploid, right? That will affect the normal life of the individual. And these beads or string-like structure in each chromatin is packed to form the chromatin fibers that are further coiled and condensed during the cell division in metaphase stage for the formation of chromosome. So packing of chromatin at higher level required additional set of protein. Now, we discussed only about what the histone protein, because it is positive in charge, but higher level of packing required another set of what you can say protein, and collectively we call it as what the non-histone protein only, that is also there in the chromosome. Non-histone protein, non-histone chromosomal protein, that is also there, but the amount is very less, and it is only for the supercoiling. Normal coiling, it use only what the histone protein to produce what the octamer. You may be heard about histone, right? A histone protein, and it is what the non-histone protein. It is role only in for supercoiling of what the chromatin threads during the cell division. And here we can see the DNA, double-stranded DNA, go for a coiling around that octamer, histone octamer, to form the nucleosome. That is what the second level. So length decrease there. And whenever this nucleosome go for another coiling, third step, again it reduce the length. Then chromatin fibers, it will be converted into chromatin fibers. Again, it go for this Cooper coiling. Then it go for the next one coiling of the chromatin fibers to form the last product, that is what the chromosomes. So chromosomes are nothing but the supercoiled DNA only. Addition to the DNA, we can find the histone protein, and we can also find the non-histone protein, particularly for the supercoiling of these chromatin fibers to produce what the chromosomes, right? Then this is what the process of what the chromosome formation. So I told you this DNA is very lengthy, having meters of length, and it is not possible to keep this entire length of the DNA in the nucleus, because nucleus is what microscopic, very small in size. Without this coiling, it is not possible to pack inside the nucleus, and this is what the process of packing. See, this is what the DNA helix, double-stranded DNA helix. Go for this coiling. This is what the nucleosome. go for coiling around this octamer. Then nucleosome go for what the repeated coiling and form what the next chromatin. And next chromatin will go for the chromatin fiber. Then chromatin fiber will go for the supercoiling with the help of the non-histone protein, and finally get what this chromosome. And such a type of, you see how it is reducing its length, how it is reducing its length, and the width of what the DNA is only 2 nanometer, whereas in chromosome it is what 1400 nanometer. 1400 nanometer. This is what the width of the chromosome. Why width is increasing? Because of what the coiling. Length is decreasing, so naturally width will increase. And this DNA, it's only 2 nanometer. Here it is what 11 nanometer. And here we can find 30 nanometer. It is 300 nanometer. And here it is what 250 nanometer. And whenever it go for coiling again 700 nanometer. And in case of chromosome, you see it is 1400, increasing the width but reducing the length, so that it is very easy to keep these chromatin threads or the chromosome inside the nucleus. This is what the way we are keeping this much length of the DNA. So tell me, actually, what is the need of such a very lengthy DNA for a cell? Basically, DNA is for the storage of information to express a character in the form of protein. See, we are secreting and producing thousands of protein in the cell, maybe in the form of enzymes or in the form of hormone. So basically, these are all protein in nature, and these enzymes only working in the cell as what the catalyst, biological catalyst, to make many chemicals to react in cellular level. Around 15,000 reactions are happening, roughly. Around 15,000 reactions are happening in the cell, and every component for the reactions are available in the cell, but it reacts only at the time of its requirement. Why? To make these reactants to react in the cell required an enzyme, and enzyme only reduce what the chemical activity, and its work as what the catalyst. So that much information should be there in the DNA. Then only it is able to produce what the different kinds of enzyme, and by using the enzymes only we will able to control all the cellular activity. If you want to make a cell alive by producing different kinds of product inside, that called as what the metabolism, required many type of protein. If you want to produce that much varieties of protein in the cell, required many information, and these informations are stored only in the DNA. That is what DNA is having this much length, and it is not possible to keep as it is in the cell, in the nucleus, because it is too small. So naturally it has to go for coiling. So if you want to express a character, what we will do? Is it possible to make what the copy of the DNA from this chromosome? No. Again, if you want to express a character by using the DNA, that particular part should go for recoiling. The way in which it is coiled, same way it go for recoiling and produce what the particular area of the DNA, and that from that area of the DNA, RNA will be copied, and by using the RNA, protein will be synthesized. But after producing RNA, again the DNA go for coiling, and this information will be kept in the chromosome. That's why I told you this genetic material should store the information related to the expression of character, and this information should be used for not only for one time. Whenever the cell required the particular protein, it should express. So that is why DNA is not directly involved in the protein synthesis. Only it is to store the information in the nucleus, and whenever it is required, it is taking that information in the form of what the Xerox copy in RNA, and use this RNA for synthesizing that particular protein with a definite chemical nature. Right? See the sequence: DNA, nucleosome, chromatin, then chromatin fibers, and chromosomes. You see, the changes happen to the DNA while coiling, repeatedly it coiling. So the chromosome is nothing but the super coiled DNA. While coiling, it's bound with the positively charged the histone protein, and for the supercoiling of the DNA into from nuclear, the chromatin threads to the chromosome required some non-histone protein also, right? Then come to the euchromatin and heterochromatin. Once this question also was asked in the examination. So euchromatin and heterochromatin. So you already know in detail about the chromatin. In some region of this chromatin, these are light stained. These are the light stained. Whenever we are applying the nuclear dye to give color to the nucleus, this area, what you can say, stained very lightly. And this chromatin is loosely packed and referred as what the euchromatin, and this area is transcriptionally very active region of a chromatin. Why it is transcriptionally very active means this area is able to produce what? RNA. This area of what the chromatin is able to produce RNA because it is very loosely packed. You can see such area here. It is what the lightly stained area. Here it is called as what the euchromatin only, and this is what the nucleosome and the octamer, and it is what the connecting linked DNA, and like that. This is what the yellow colored one, we call it as what the DNA only, and it is not very tightly packed, very loosely arranged. And such a type of area of the chromatin absorb color very lightly. Whenever we stain the chromosome, this area stain very lightly. And this is called as what the euchromatin. And this area is very active in transcription. Transcription means what? The process to produce what? The RNA from the DNA. And this area is able to produce what? The RNA from the DNA because it is arranged very loosely, not much coiled. Very loosely packed, so that this area is what? The transcriptionally very active. Whereas another one, that is what the heterochromatin. Whenever we give color stain to this chromosome, this area get more stain, and these are what the dark stained area. But it is very inactive transcriptionally because these are all what connect each other, and arrangement is what very close to each other, and not very loosely packed, so that this area, getting the DNA back and go for the recoiling and production of what this RNA from DNA. It's very difficult, and this area normally stained darkly because it is very closely packed, absorb more stain, darkly stained area, and it is very, very inactive transcriptionally. The transcriptional process is very slow, this area. So that is called as what? The heterochromatin area. Some region of chromatins are loosely packed, and this area is absorbed only little stain, and it is called as what? The euchromatin. As I told you, this area is very transcriptionally very, very active, whereas other areas of chromatins are densely packed, very close to each other, connected very tightly, and such a area absorb more stain and darkly stain, and it is called as what? The heterochromatin area only. And this area is transcriptionally very, very inactive because these are all densely packed, very difficult to recoil the DNA from it, and very difficult to what? Go for the transcription, and we call it as what? The heterochromatin area, right? And this question asked once in the board examination. Then come to the Griff experiment. This experiment is to prove that genetic material is not protein. While discussing previous slide, I told you only the DNA or RNA can act as a genetic material, because genetic material should have some kinds of basic properties like mutation, then store information, or what you can say multiplication. Such a type of things are not there in case of protein. Only what you can say genetic material is or RNA. Genetic material should be RNA or DNA. That is what the nitrogenous base, not protein. But basically, DNA is better than what RNA, because chemical nature of RNA is not good for storing the information. But this DNA is evolved for the storage of information or for storage of, expression of information or new character only. So there is an experiment conducted by the group in 1922. His name is what, the Frederick Gruft only. We call it this experiment as what, the Gruft experiment only. And this experiments to prove that genetic material is not a protein. It's not an RNA, but the genetic material is DNA only. As I told you years back, conducted in 1922, right? And during those period, there was a big controversy between the scientists, which is what the real genetic material express a character in the individual. Some are telling it's protein, some are telling it's RNA. And because no, we don't have much idea about all these biomolecules during those time. And this Griffith conducted one experiments to prove that protein is not a genetic material and RNA is also not a genetic material. Their real genetic material is what, the DNA. And this experiment only we call it as what, the Griffith experiment. In 1928, the Frederick Griffith conducted an experiment in Streptococcus pneumonia. It is a pneumococcus bacteria, a cocci bacteria, round-shaped bacteria, cause a disease called as what, the pneumonia. And cause a disease called a pneumonia to prove the transforming principle. From this, what the experiment only, some other scientists prove this transforming principle. We will see the principle later. See, when Streptococcus pneumonia are grown in a cultured medium, some produce a smooth, shiny colony. It is smooth. We call it as what, the S strain. S means what, the smooth strain only. Surface of what this coccus bacteria is very smooth and it is shiny. And another variety is what, the rough colony only. So while cultivating Streptococcus bacteria in a culture medium in this laboratory, I found two strains. One is very smooth and another one is what, the rough. The smooth one with a shiny surface, we call it as what, the smooth strain, and another one is what, the R strain. S strain is smooth externally because they have a mucal coat. Why it is shiny in external appearance? Because it is having what, a mucal coat. We call it as what, the polysaccharides only, while R strain does not have. R strain is rough because they don't have such a what you can say, smooth mucal coat on its surface. That polysaccharide is not there. And Griffith conducted the experiment with Streptococcus pneumonia and mice. You may be remembering the experiment conducted by the Griffith. He used two types of strains of this coccus bacteria, Streptococcus pneumonia, and he used what that mice also to do his experiment. Let us see the simple experiment conducted by the Griffith. Simple experiment only. A strain, it's having what you can say, a smooth surface, and it is what, virulent, can cause disease. So he collected the strain and injected directly into the body of mice. Naturally, that mice died due to pneumonia. Then he injected the R strain also in the body of another mice. Their mice lived because it is non-virulent only. Concept is very simple. There are two strains of what, the coccus bacteria, pneumococcus bacteria, and it is causing what, the disease. And he used these two strains and injected in the body of the mice, S strain and R strain. He found S strain is what, the virulent, and because of its injection, the S strain, that mice died. Whereas R strain is non-virulent. After injecting the R strain, rough strain, pneumococcus bacteria into what, the body of the mice, that mice lived because it is non-virulent. Then he injected S strain again, but after killing by using heat. Heat killed S strain because it is already died. This S strain bacteria is already died. Of course, it is virulent only, but after what you can say, heating, he made it what, he killed this S strain and injected into the body of mice, and where that mice lived. Natural, because it is what, the dead body of the S strain only he injected in the body of the mice, and it's naturally is non-virulent. Then later, he mixed that he killed a strain. It is what the non-virulent only, and also mixed with what you can say R strain. That is also non-virulent only. He mixed these two. Heat killed strain, it's non-virulent, and R strain, that's so non-virulent. And this mixture he injected into the body of the mice. What happened? That mice died. And he recovered the living strain bacteria, then collected the blood sample from the bacteria and identified. And he found that living strain bacteria from the dead body of the mice. Living strain. Actually, he injected only what the dead body of the strain, heat killed strain, and mix it with the R strain. Then, but he recovered the living strain bacteria from the body, from the blood of these mice. How is it possible? How is it possible? He injected that living R strain, non-virulent only, but he found what the strain, that living strain bacteria from the blood of these mice. What is the reason for that? Simple. This R strain bacteria is living and S strain bacteria is heat killed. Whenever he mixed this, what the heat killed S strain with R strain bacteria, some of the material present in the S strain can produce a character, a genetic material. It transferred to what the R strain. So because of the transfer of the genetic material from the heat killed S strain to R strain, naturally that will be converted into what? S strain. S strain means what? A virulent only. That only killed the mice here. This is what the simple experiment conducted by the group. We have to explain the reason for this transformation. That's why I told you this is what the transformation principle. Why it is called as transformation? Here, R strain is transformed into S strain. What is the reason for the transformation of R strain into S strain? It is because of what? The passing of some material from the heat killed S strain to the R strain only. You know, the genetic material is stable. That is there in the S strain even after killing this bacteria. So whenever this heat killed S strain mixed with the R strain, the material, the real genetic material can express some character in the individual, pass from the S strain bacteria to R strain bacteria. That material transferred from the S strain to the R strain only convert the transform this what the living R strain bacteria into what the S strain bacteria by producing what that caught. So it became smooth, and such a type of living S strain bacteria only he found from the dead body of this mice. That is why we call it as what the transforming principle. Some genetic material taken from the S strain, heat killed S strain, convert it into what the taken to the R strain, that only transform the R strain non-virulent bacteria into what the S strain virulent bacteria. That is why that mice died due to what this pneumonia. So this is what the GIFT experiment. This rough strain, it's a non-virulent bacterial colony injected in the body of this mice. So result, mouse found very healthy. Then smooth virulent, it is can cause diseases, and it's injected directly in the body of the mouse. Naturally, these mice die due to the pneumonia. Then heat killed the smooth virulent strain that injected in the body of this mouse. So what happened? This mouse lived. Why? Because it is heat killed only, unable to create pneumonia in the body. Then he mixed both this rough, but you can say non-virulent strain. You know, that is what non-virulent only cannot cause disease in the individual, and this is also heat killed only. Right, that is what heat killed only. Mix these two strains together and injected in the body of this mouse. So this mouse dies. And see, he collected the blood sample from the dead body of this mice and analyzed. he found the living strain bacteria in the blood sample from the dead mouse. He didn't inject that what the living strain. He killed strain only he injected. And the living bacteria is what the non-virulent that R strain only injected. But here he found the living dangerous strain bacteria. Why? Because rough bacteria strain is living. So from this heat killed bacteria smooth strain, some material taken by the rough strain of this bacteria, and this material only making this rough bacteria into what the active one. So this is what the experiment conducted by the group, and we will see what the transforming principle and chemical nature and other related experiment conducted by the other scientists. So we will see it after a break, and we will meet again afternoon from 2:30 and 2:30 to 4 o'clock. One section will be there as usual only, and evening also you will have a section 6:30 to 8 o'clock, right? So there we will discuss in more detail about what the characteristics of the DNA and relevance of this Griffith experiment. So now you can go for your breakfast and come back at 2:30. We will discuss some more detail related to the topic. Then bye. Thank you so much.